Thank you, Colleen. Hello, everybody. As Colleen said, my name is Jennifer Polson. I am a master's candidate in Dr. Ping's lab in the Department of Physiological Science, and I will be presenting my thesis work up until this point and give a brief overview. Let's do that. All right. Cardiovascular disease remains the leading cause of death among populations in the United States. This high rate of mortality may be attributed to irreversible deterioration of cardiac function caused by long-term molecular alterations in cardiac tissue, which can preclude the development of effective therapeutic strategies. The manifestation of pathological phenotype is very complex, as there are multiple dimensions of information that underlie cardiac disease pathology. Omics technologies provide the opportunity to acquire high-throughput data and comprehensively profile a physiological phenotype within one dimension. One such dimension to use is proteomics, in assessing the entire range of proteins that occupy a cell. And there are multiple ways to assess the proteome. The rate of synthesis and degradation, or turnover, illustrates one novel facet of the complex factors that govern a phenotype. Protein turnover is governed by synthesis and degradation machineries. Synthesis machineries, primarily consisting of the ribosome's large and small subunits, are transcriptionally regulated. Degradation machineries consist of the proteasome system, specifically the core particle and the regulatory particle, as well as free-floating proteases, peptidases, and the autophagy lysosome system. Sorry about that. So the degradation of unfolded proteins requires the coordinated regulation of these systems in parallel, but my study specifically is going to focus on the ubiquitin proteasome system. The ubiquitin proteasome system requires the functional assembly of the proteasome in its entirety, which is composed of two types of units. There's the core particle, or 20S unit, which has 4 stacked rings that form a central pore. Each ring is made of 7 proteins. There are 2 inner rings that are made of beta subunits, and they have the active degradation sites. And then there are 2 outer rings with alpha subunits that maintain a gate for the protein to enter. There's also the regulatory particle, which recognizes ubiquitin tags attached to proteins and initiates the degradation process. In the past, proteasomes have been targeted to assess cardiomyopathies. Several studies have demonstrated the efficacy of proteasome inhibitors for preventing or alleviating symptoms associated with cardiomyopathies in animal models. One example is bortezomib, which has shown potential for restoring cardiac function in patients with restrictive cardiomyopathy. However, recent data indicates that inhibiting the proteasome leads to an accumulation of misfolded proteins in cardiomyocytes and can activate apoptotic signaling pathways and cell death. Indeed, proteasome functional insufficiency is directly linked with proteinopathies such as Desmond-related cardiomyopathy. Therefore, a more sophisticated therapeutic approach may be to target specific components of the ubiquitin proteasome machinery. And that's what leads me to my research question, which is, how does the turnover of proteasomal machineries vary in cardiomyocytes under stress conditions? In order to assess this, I used a reference data set produced by Edward Lau, as well as other colleagues in my lab. The data set is seen in the publication, a large data set of protein dynamics in the mammalian heart proteome. Briefly, I will go over the system and model that they used. Prolonged stresses on the heart can induce many forms of pathological conditions, including cardiac hypertrophy, which can precede or exacerbate severe cardiac events and critically inhibit successful cardiac outcomes. Hypertrophy is defined as an adaptive response to pressure or volume stress, mutations of sarcomeric or other proteins, or loss of contractile mass from prior infarction. And hypertrophy can be considered adaptive in a physiological sense, such as an exercise-induced hypertrophy, or pathological, which is called maladaptive cardiac remodeling. Isoproterenol is one way to induce hypertrophy in the heart. It is a non-specific beta-adrenergic agonist and stimulates the adrenergic response in the autonomic nervous system. Isoproterenol binds to beta-1 adrenergic receptors in the heart muscle, and it increases the heart rate and contractile force. Over time, this can lead to maladaptive or pathological hypertrophy and eventual heart failure. The beta-adrenergic signaling pathway is shown to be involved in hypertension, heart failure, heart attack, cardiac stress as a compensatory function. It's important to note that the dose of isoproterenol is, in this study, is low enough to induce hypertrophy without inducing immediate heart failure, and therefore allows us to ascertain the effects that are happening. To generate this data set, 6 genetic mouse strains were chosen for their variable susceptibility to beta-adrenergic stimulation to create a diverse genetic cohort. For every experimental group, the following protocol was implemented. Every mouse in the study received deuterium oxide, or just heavy water, as a form of tagging the actual protein turnover. They received 2 IP injections and then received dose drinking water for the remainder of the study. In addition, mice under ISO conditions received a mini osmotic pump with 15 milligrams per kilogram per day of isoproterenol to induce hypertrophy. For this study, mouse heart samples were collected at the following time points listed on the slide, and these samples were homogenized, and the turnover was determined via mass spectrometry and methods developed in-house, including the software ProTurn. And we were able to generate the turnover rates for proteins under control and ISO conditions. For my study, I chose to select a few protein groups, one such as the proteasome subunits. This is a total of 50 were identified in the data set. For the purpose of comparison of other turnover machineries, I chose to also extract non-proteasomal degradation proteins as well as ribosomal proteins, of which there were 40 and 76 identified in the data set respectively. And finally, to observe downstream effects of this, I chose to assess contractile or sarcomeric proteins from a selected list, of which 60 were identified in the data set. For each group, I assessed all 6 strains, and the result is a plot matrix that contains 24 panels. So in each panel, I plotted the turnover rates for a protein group. An individual dot is an individual protein's turnover rate under control and ISO conditions. The x-axis corresponds to the control half-life in days, and the y-axis corresponds to the ISO half-life in days. Now, there are biological implications that exist based on where a point lies on its plot. For clarity, I added a 0 alteration line, which shows if a point is not substantially altered, it will lie very close to this line, and that's scenario A. In scenario B, a protein would have decreased half-life. It would lie on the bottom right half of the graph, and that would mean increased turnover. And in scenario C, if it lies in the upper left part of the graph, it will have increased half-life and therefore decreased turnover. And these plots allowed for visualization of protein turnover distribution. Now, from these plots, I also established metrics that allowed me to assess and compare between the strains. The first one is the polygonal area, which is just an area of a polygon drawn around the plotted points of the functional cluster, as you can see on the graph. The second one is the Euclidean distance, which is the mean value, the absolute distance of each protein from that 0 alteration line. And finally, the directional index, which is the relative amount of proteins with increased turnover versus decreased turnover, and this varies from -1 to +1. These 3 metrics have biological implications that we can interpret based on the values. So the area represents the 2-dimensional range that the specific functional group occupies. This biologically indicates intrinsic synchronization of a protein group, and therefore the regulation of that group. And just for clarity, a decreased area would imply increased synchronicity and therefore increased, tight regulation. Distance shows the level of alteration of a protein group, indicating the susceptibility of the group towards isostimulation. As one might expect, the larger the distance, the greater the sensitivity of a protein group. Finally, the directional index is a measure of the direction of modulation, which elucidates the trend of protein turnover alteration under ISO conditions. As I said, they vary from -1 to 1. A value of -1 means all of the proteins turn over more slowly. A value of +1 shows that they all turn over more rapidly. And a value close to 0 kind of indicates more of a divergent or balanced turnover modification. So with these metrics, I created the 24 plots just to show briefly what these kind of look like visually, so that I was able to see what was happening within each protein group along each strain. And I also created the metrics for each of these plots. So on this table, the I indicates the index, the D indicates the distance, and the A indicates the area. And I will talk about the brief biological implications of these different metrics. So first I chose to look at the proteasome machineries. And the proteasome is unique in that across the 6 strains, proteasomal modulation is less than that of other protein groups, and it occupies a lower average range, which may indicate more synchronicity or regulation. Among the specific strains within this, it became apparent that ISO induces greater modulation among C57 and DBA, with C57 having a divergent response and DBA showing a concerted decreased turnover. CEJ was actually the most resilient to ISO-induced stress and exhibits the highest synchronicity. The last 3 strains, FEB, AJ, and BALB-C proteasome subunits are moderately responsive to ISO, although they do exhibit different trends. FEB has concerted increased turnover, AJ has decreased turnover, and BALB-C has a divergent response. Among the 6 strains, this shows that this is a genetically heterogeneous population. And in choosing to see that there was a diverse population there, I looked at the distribution of all of these across all 6 strains. So this panel A is a violin plot, which shows the distribution of protein turnover under control and beta adrenergic stimulation via isoproterenol in blue and red respectively. The distribution of proteasomal turnover is laid over the distribution of the entire proteome just to understand it for comparison. So it's apparent in this that you can see that the proteasomal ranges occupy a much narrower space than do the entire proteome. To account for differences in protein pool number, these are by density rather than absolute count. Finally, the middle lines indicate the median of each of these distributions, with the green representing the control median and the yellow indicating the ISO median. Panel B is a volcano plot. And what I chose to do is for every proteasomal protein, I calculated an average fold change across all 6 strains and plotted the log2 fold change on the x-axis and the p-value of the value of this approximation of the 6 strains on the y-axis. I applied the following statistical threshold of p has to be less than 0.05 and a differential threshold of a 20% fold change. And as a result, 2 proteasomal proteins were identified, PSMA5 and HSP90AB1. So PSMA5, otherwise known as the proteasome subunit alpha type 5, displays an increased half-life and decreased turnover. It is a 20S catalytic particle component, and it is responsible for the first step of proteasome complex assembly. It serves as the binding site for chaperones during the mature alpha ring formation. As far as disease is concerned, PSMA5 is particularly susceptible to many post-translational modifications, including acetylation and phosphorylation, and gene regulation under cellular insult. Interestingly, recently, it has been shown to display endoribonuclease activity and is able to degrade the mRNA of regulatory transcription factors such as c-Myc and c-Fos. Overall, this indicates that PSMA is a sensitive indicator of proteasome function across multiple dimensions of molecular profiles. HSP90AB1, otherwise known as heat shock protein 90 beta, has a decreased half-life and therefore increased turnover. It is a chaperone protein that binds to other proteins and stabilizes them during varied points of the protein life cycle. In the case of misfolded proteins, HSPs mediate protein degradation and can be involved in cellular localization. Clinically, they have long been identified as having upregulated gene expression in cancer cells, and it binds extracellular to TGF-B beta receptor 1 to induce TGF-B beta mediated collagen synthesis in fibroblasts found in the heart. Finally, it has been shown to be cardioprotective in hibernating animal models against ischemia reperfusion injury. So I also chose to use this workflow to assess contractile proteins as well. Contractile cluster as a whole has a larger range in both experimental and ISO conditions, but also displays minimal modulation comparatively speaking to other clusters. Among the specific strains, there's unique trends that are not seen in proteasomal subunits. So the contractile units that are most susceptible are in the CEJ, DBA, and BOLB-C strains. There's decreased turnover in CEJ and DBA and enhanced turnover in BOLB-C, which you can see on the graphs. AGA contractile proteins are the most resilient and primarily exhibit a slower turnover among subunits. And finally, C57 and FEB exhibit moderate divergent induced turnover modulation. And I then again continued my workflow. So panel A is again the violin plots of this. It's apparent on here that the distribution of contractile proteins is a much larger range, as I said before, than was the proteasomal machineries. And finally, I applied the volcano plot that is same with the same thresholds that I did before, and I identified 10 proteins whose turnover is significantly modulated as a result of beta-adrenergic stimulation. From a functional standpoint, these contractile proteins span multiple compartments within the sarcomere, including the thick filament, in which there are different myosin light and heavy chain proteins identified. Within the thin filament, troponin I and troponin C were found to be differentially modulated. In addition, there were proteins identified in the Z line, in the desmosome, in the gap junctions, and in the costamere. So all of these occupy different functional regions that enhance contractile and are responsible for contractile force within a cardiomyocyte. To summarize, we were able to inspect the turnover features for selected protein groups and show that they have unique features among each strain. And this also shows that there were distinct modulations within a protein group as well as within a genetic strain. We were able to determine the level of turnover modulation among specific proteasomal subunits and identified the two proteasomal subunits, the heat shock protein 90 beta and PSMA5, as significantly altered. We were also able to identify signatures of contractile protein turnover alteration, with the following proteins being upregulated and downregulated, specifically in regards to turnover. Finally, this workflow was developed to analyze protein turnover and add another dimension of data to study the manifestation of molecular changes into pathological phenotypes. And this workflow can be applied to other proteins very easily. In the future, as this only provides one dimension of data, there can be multiple ways to characterize the proteasome using multiple parameters. We can compare the basal expression of proteasomal components for 6 genetic strains and compare the proteolytic activities for these mouse strains after 7 and 14 days of ISO treatment with the basal conditions, and examine the differences in complex assembly and individual proteolytic functions. To achieve a more holistic understanding of the degradation as a whole, we could expand this to other degradation machineries, namely the lysosomes and free-floating proteases. We can conduct proteolytic assays for both lysosomal and free-floating protease degradation and isolate mitochondrial fractions to determine expression of protease components and proteolytic capacities. With characterization, quantification, and functional data, this would provide multiple dimensions of information to decode the molecular foundation of protein degradation dynamics during cardiac stress. Briefly, I'd like to acknowledge that everyone in my lab the people in my lab who were crucial to helping me develop this project and bring it into fruition. Everyone has been really helpful in helping me understand all of the technical details and the physiological details underlying this data set and their implication. And thank you. I appreciate your time.
Thank you, Jennifer. Do we have any questions?
This is, sorry, go ahead.
Go ahead.
So it's Pei. I just have a comment. I think, Jennifer, this is honestly not buttering you up at all. The best master thesis I have ever seen, with respect to careful work, clarity of presentation. And so I want to say congratulations.
Thank you.
And I appreciate the careful analysis of one specific functional subgroups. And I have a couple of recommendations. One is, I know you and Dean have been working very hard to update the GeneWiki page on this particular group.
Mm-hmm.
And so if the things that you have been able to dig out and translate or extract the knowledge from the data sets Edward and Travis have created, I think the GeneWiki is the grand opportunity for us to do it, right? You spent all these efforts, and that analysis was very detailed. And I don't know if, Gia, do I have you on the call? Gia? If I don't have Gia, Travis, are you there?
Yes, I'm here.
Wonderful. Can you work with Gia to follow this workflow that Jennifer has created to work on the stress-sensitive protein subclusters?
Sure. I'll talk to Gia to use this workflow to focus on that.
Thank you, Travis. And Andrew, sorry to interrupt. You had a question or comment?
Yes, I did, but no apology necessary. I agree, Jennifer. Great work. Congratulations. I had a question about, and I like all three of your quantification methods of the plots. It occurs to me, though, especially from seeing some of the plots, that your area calculation is actually pretty sensitive to outliers. There are a few cases where you saw one protein that was way out there, and then your polygon has to reach out there, and that drastically increases the area. Have you looked at measures of area that are less sensitive to outliers?
So one thing that I did apply, I don't have it on this data, is I looked at the rate at which the error of the turnover rate was too high, and that actually usually accounted for the area of the outliers, because that meant that there was a certain rate of variation that could be accounted for. And when I accounted for that, there was less sensitivity to outliers. I think you're specifically talking about a couple, like, for example, the DBA plot, the top proteasome one, there's actually one protein that was just totally out of whack. So that would be another way. And that was one way. If you have a suggestion for a different way to calculate it, though, I am all ears.
There must be a way to do it based on, you know, percentiles. I can't think of it, but I can imagine that it exists.
Yeah.
So yeah, bottom line, I don't have anything actually useful to say to you, except I think it would be that my strong suspicion is that algorithms out there exist.
Yeah, yeah, okay. I'll definitely look into that and maybe make it density based or something like that to assess for area that doesn't account for, or that will account for the outliers. Thank you very much for that feedback.
Yep, and I have a second question too. So so clearly you've applied this to turnover and half-life. You know, is there any reason that you think your method wouldn't also be applicable to sort of set analyses based off of abundance, not only for proteomics but for transcriptomics?
I mean, that is definitely something that I feel like would be valid for comparison, because this is just, you know, a measurement under two different conditions. So anything could be applied. I think for expression or abundance, you would definitely want to apply different statistical thresholds than 20%, because the rate of turnover, you know, assessing a change in rate is like the acceleration. So the differential threshold is a little bit lower, but I think you could definitely apply this to any sort of data under which you would have two values.
Makes sense. Then my last request would be, you know, I think you should post the code somewhere on GitHub so that
I do have it on GitHub. Thank you. Yeah.
Fantastic. If you could just email me that, I wouldn't mind taking a look.
Yeah, definitely. I will do that.
Thanks. Nice presentation.
Andrew, to add to Jennifer's answer regarding your question on protein abundance as well as mRNA. So thanks to Travis and Gia, we've been fortunate working with Dominique to have actually a new set of protein abundance evaluated by different tagging methods. And so that's an independent measurement of just protein abundance derived from deuterium labeling. We did that because that was what Nature Communication wanted. So unfortunately, when Jennifer was working on her thesis, the data was not available. It just became available less than a few days ago. We did manage to put all that new data into this resubmission to Nature Communication. I don't know if that would be sufficient to please the reviewers, but let's hope if it doesn't go well there, those data will get published somewhere. But maybe someone else can continue Jennifer's effort to do these analysis on the absolute quant for the abundance, as well as for the transcriptomics data that we have collaborating with Jake and Yibing.
Great, makes perfect sense.
Do we have any more questions for Jennifer?
Hi, this is Wei. Well, this is a great presentation. I really enjoyed it. I just have one suggestion for potentially to address Andrew's first question about this area and the sensitivity to outlier. I wonder whether it would make sense to do a mixed Gaussian model to fit this distribution, and then later you examine maybe the differential pattern of those learned Gaussian mixed Gaussian model.
Thank you.
In addition to just look at the area.
Yeah. Thank you very much for that suggestion. I appreciate it.
This is why I'm glad we have smart people on the call.
Oh, just a random thought. I'm not sure if this works, right? So someone, may want to try that. I'm glad to talk, more in person.
Okay. Thank you very much. I appreciate that a lot.
So Jennifer, I'm going to count you to talk to Wei to get help.
Mm-hmm.
And that'll be really cool.
Yeah, that sounds great.